Okay, so very good evening to all. Okay, so I welcome you all for this online session. So this is my second session. Okay, after 6th September. Okay, so in the previous session, we have discussed about the biodiversity, on what are the biodiversity and what are the different types and sources, and then the values of biodiversity. So today, so we are going to discuss on the factors which are endangering the biodiversity. Okay, so the causes which will the reason for the loss of biodiversity and the various threats to the biodiversity. Okay, and then hotspots, what are hotspots, and then biodiversity and the conservation aspects. So that's what we are going to discuss. So actually, what threatens the biodiversity? So normally, many human activities, they have a negative effect on the biodiversity. Each time we change the natural environment, we change the environment that biodiversity lives in. Actually, the species extinction and habitat loss, okay, which is one of the cause of the threats to biodiversity. And then today, the biodiversity loss is 1,000 times higher than the rates of that is found in the fossil record. So the causes of the biodiversity. So if you discuss the causes, so previously it was known as urbanization, agriculture, water diversion, that means the reservoir creations, okay, recreation and tourism development, pollution, ranging, all these things considered as causes. So if you look at the threats to biodiversity, you can segregate them into different factors. One, the habitat destruction. Okay, so habitat destruction, you know very well. That means the source, that means the biodiversity rich areas. Okay, so they have destructed either by deforestation. Okay, so loss of the habitat is the mainly caused by the deforestation activities. So the forest and grasslands have been cleared for conservation into agricultural land or settlement areas or to make some developmental project. So the forest and grasslands are the natural homes of thousands and thousands of species which disintegrate due to the loss of their natural habitat. And even you can further subclassify the same into habitat fragmentation. So what is habitat fragmentation? Sorry. Uh, so Kanivanan requested that to make it as a full screen mode. I have tried, but once I make it as a full screen mode, my slide is not moving, Kanivanan sir. That's why. Okay, so from next class, I will try to change it, because if I make it full screen mode, my slides are not moving because parallelly I'm watching in my mobile. So that's why I have put it in the normal mode. Okay, kindly excuse me this time, and then next time I will try to rectify. So the habitat fragmentation, sometimes the habitat will be divided into small or scattered patches. So many causes are there, mainly due to the natural calamities. Okay, so this may happen, and this phenomenon is known as habitat fragmentation. So due to this, many wild animals, okay, and then some birds and even plants or get vanishing. So next is the alien invasive species, non-native species invasion. So this may be the another causes. And then genetic pollution. So what is genetic pollution? Genetic pollution is a controversial term actually. So for uncontrolled gene flow into a wild population, it will happen when the uncontrolled gene flow into a wild population. So it's also defined as the dispersal of the contaminated, altered genes from genetically engineered organism to the natural organism. Okay, so if it is in plants, it may be due to the cross pollination. Okay, so this is called a genetic pollution. So this is another factor. And then over exploitation.
Sir,
Yeah,
hi sir, I'm Shweta.
yeah,
Sorry to interrupt you, sir. Can you just explain this genetical pollution once again?
yeah. So actually, so what is genetic pollution? Actually, I told you the uncontrolled gene flow into a wide population. So this may happen. Okay, so you know what is the reason behind this? Okay, so the genetic material, okay, which can be unintentionally introduced into a population. Okay, this is called genetic pollution. So this will.
Any possible example, please, which we
Hello, sir.
can, so that it is easy for us to understand?
Sorry, I'm not able to hear you.
Yeah. Any possible example which we can share with us so that it is easy for us to relate to it?
Examples. You are asking for some examples, sir.
Yeah, so
Yes.
for
Yeah.
genetic pollution, GM crops can be considered, sir?
Yeah, GM crops. See, if it is plants, you can consider the GM crops, and then if animals, you can. So one good example, many fishes you may know that. Okay, so which is by intentionally or non-intentionally, which is introduced into your normal ecosystem. Okay, the fishes, okay, which are created for the commercial purposes. Okay, which may
Exactly.
be which may be released into the environment. So now I hope you may got it right.
Yes, yes, perfect, perfect, sir. Yeah, very clear. Fishing, we can share an example that will be Paku or the Piranha, which has been introduced into Indian waters today.
Yeah. So, like that. So I excuse you. Can you please keep your questions in mind, and then can you please ask me at the end of the session? Okay.
Sure,
So
sir.
this will be convenient for me, right? So, so now someone requested to get in. Okay. Now, so next to that, over exploitation. So you know the reason. So the over exploitation is mainly due to the human activity, climatic change, and then even diseases. So what if you see this? The I have listed some of the factors which are affecting the biodiversity: the construction of dams in forest areas, industrial waste, urbanization, use of some insecticides and pesticides, and poaching of wild animals, over exploitation of the natural resources, and degradation of the habitats, oil spills, and discharge of effluents, whereas in the marine ecosystem is concerned, which will affect the marine biodiversity, global warming, ocean depletion, acid rain, everything. All these are some of the factors which will affect the biodiversity. So we can classify them into many factors. Okay, so you know one thing, the man-wildlife conflict. Okay, the man-wildlife conflict, when it will arise, when the wildlife start causing the immense damage and danger to the man. You know, so in this condition, it is very difficult for the forest department to compromise the affected villages and to gain the villages to support for the wildlife conservation. So many examples we can say, you know very well. Okay, so long back in Sambalpur, Orissa, okay, more than 195 humans were killed in the short period of 5 to 6 years by the elephants. So that to retaliate the same, what the villagers they have done, the villagers have killed nearly 95, 98 elephants, and even 30 elephants are were badly injured. So, and one more thing, in Samrajnagar, Mysore, okay, several elephants were killed because of the massive damage done by the elephants to the farmers' cotton and sugarcane crops. Okay, so like that, many examples are there. Sometimes some the villagers sometimes they may hide some explosives in the sugarcane field, which will explode when the elephants enter into that field. Okay, and then so the man-eating tiger, which will kill nearly 16 Nepalese people and one 4-year-old child. Okay, in a Royal Chitwan National Park in Kathmandu. These are few examples. Okay, so what are the factors which will influencing the man-animal conflicts? Okay, or which will causes the man-animal animal conflicts? The shrinking of the forest cover, which will compel the wildlife to move outside the forest and to attack the fields and humans. So the human encroachment into the forest area, which will induces a conflict between man and the wildlife. Okay. So what the injured animal? Okay, usually they have a tendency to attack man, and especially the female wildlife. Okay, which will attack the human if she feels that her newborn cubs are in danger. Okay, normally you can see it in dogs, even in our domesticated dogs also you can see this. When it has given new young ones, if you go near that, and then it will try to bite you or so bark at you. So earlier the forest departments they used to cultivate the sugarcane, paddy, coconut trees in the sanctuaries. When the favorite food of elephants are not available, they feed them to the field. Okay, but now due to the lack of such practices, the wild animals move out of the forest for searching of food. Often, even some villages put electric wiring around their crop field. Recently, near Coimbatore also, so we have witnessed a lot of elephants get died due to this. Okay, so all these are maybe the causes of the conflict. Okay, so what may be the remedial measures? Okay, so adequate crop and cattle compensation schemes must be started. So the solar-powered fencing must be provided along with some electric current-proof trenches, which will to prevent the animals from entering into the field. And then the cropping pattern can be changed near the forest borders. And then adequate food and water should be made available for the wild animals within the forest zone. Okay, and then so development and constructional work in and around the forest region must be stopped. Okay, so these are the thing. So here, if you take it as a habitat loss, the thinning and fragmentation, as we discussed, the thinning of the habitats, that means the reducing the size of the habitat, original habitat, and fragmentation, and then the destruction of an ecosystems, and then ecosystems components like the plants, soil, and then hydrological and nutrient resources. So actually, if you look at that, these are some primary drivers, okay, for the biodiversity loss. And if you look at the influences, okay, the human population growth, and then the increase in consumption, and then reduce the resource efficiency. So all these are considered as influences. So as I told you, the invasive species, a non-native species that significantly modifies or disturbs the ecosystem it colonizes. And then overexploitation, the process of harvesting too many aquatic or terrestrial animals, which will deplete the stocks of some species. And then pollution, the addition of any substances or any form of energy to the environment at a rate faster than it can be rendered harmless. Climatic change, which is associated with the global warming. So modification of Earth climate associated with rising levels of greenhouse gases in the atmospheres. Okay, and then this may be another process. So what will happen if the biodiversity get lost? So the reduction in the number of genes, the individual organisms, and then species and ecosystem, which is in the given area. So as I told you, the construction of dams, the other causes, and then illegal lodging. Okay, so the rising population. So the population, which is keep on rising, growing human population, which needs more space for living and for agriculture and infrastructure, which leads to encroachment in the forest area. And then deforestation, stripping forest and greenlands for the infrastructure development or for commercial area development. And hunting and poaching. So this is the wildlife is hunted for the products. That means either for the skin, for meat, or for any other purposes. So, and then mining activity. So the mining, creation of mining like coal mining, marble, and gold and copper. Okay, so just for the resources, mineral resources. So the creation of the mining. in these areas. So then impact of the exotic species. Okay, and then the climate change, natural calamities like floods, droughts, forest fire, okay, and then volcanic eruptions, and so on. And then overuse of the natural resources. That means overconsumption of the fossil fuels, and then the energy resources. So all these things. So, so these are the causal factors. Okay, the population, consumption, and then the resource efficiency. So there are some indirect drivers. Okay, so like you may classify them into uh, agriculture and forestry, fishing and hunting. Okay, so all these things. So what have some direct pressures on biodiversity and ecosystem? Okay, so they have some pressure like their loss of habitats, fragmentation of their habitats. Okay, so and so on. So the state of the global biodiversity. So the benefits that the people obtain from the ecosystem. So you can classify them okay into different uh, ecosystem: terrestrial, freshwater, marine, and so on. So the main cause of biodiversity loss. Okay, so more than 16,000 species are threatened with extinction. Okay, so you know all the reasons are given. We discussed now. Maybe it may be due to global warming, or habitat destruction, or overexploitation, pollution. So examples also I given here. Okay, so you look at this. Okay, so I just want to give one example here. For this thing, just go through this. So next is, so not only in terrestrial region, if you go to marine, okay, now by it was expected that okay by 2050 there will be more plastics in the ocean than fish. So this is coming under the causes of factor, the pollution. So the biodiversity loss of plants and animals due to the overharvesting, that means overfishing, okay, and then due to the habitat loss, and for commercial markets, they're exploiting it, animals from marine region. So all these things, okay, are the factors. So the known cause of animal extinction still since you can see this. So the species intervention is 39% and habitat destruction is 36% and hunting is 43%. So we classify them into the threats in terrestrial areas: degradation, destruction, fragmentation of the natural habitat, decrease in the capacity of the agricultural areas, pollution of the soil, air and water, invasions by the alien species, non-native species, and then epidemics affecting the wildlife, climate change, pollution and eutrophication, desiccation of soil and wetland, recreation and leisures, and then if it is in marine areas, overfishing, pollution and destruction of the seafloor, alien species introduction, and so on. So, so what we are going to do for that? So this will cause some species of endangered. So there, if you look at that, a species which will be said to be endangered when its number has been reduced to a critical level. Unless it is protected or conserved, it is in immediate danger of extinction. So if you go through the data in India, more than 500 plant species have been identified as endangered species, and more than 110 mammals and 160 birds are estimated to be endangered species. But the India's biodiversity is threatened due to the habitat destruction, degradation, and the overexploitation of the resources. So if you look at that, some important endangered species, a few species of endangered or reptiles, mammals, birds, plants. Okay, so if you take in birds, the peacock, Siberian white crane, pelican, Indian bustard. In reptiles, tortoise, green sea turtle, python. If you take examples in mammal, the Indian wolf, red fox, sloth bear, okay, so on and so on. Even in plants also, a large number of medicinal plants, okay. And then other than the medicinal plants, we have sandalwood tree, like santalum, okay. So these are some examples. So if you look at the factors which are affecting the endangered species, so pollution again. So the humans dispose their waste products, okay, on nature. So either on land, river, okay, and then air is get polluted severely by the human action, and then these pollutants, okay, enter the environment, and they will travel through the food chain, and it will accumulate in the tissues of the living things, and finally, which will leads to death. And then over exploitation. So the over exploitation of the natural resources and poaching of the wildlife also lead to the extinct of wild animals. Okay. So as discussed, climatic change. Okay, all these are some examples. So what are the remedial measures? Okay. So actually, the international trade in endangered species of wild fauna and flora. Okay. So so what they have made an agreement between the governments to aim that to ensure the international trade in specimens of wild animals and plants to okay, which does not threaten their survival. And then several international conventions which helps to protect the endangered wild species. So among this, one of the most reaching treaty is the Convention on International Trade in Endangered Species. Okay, so this is signed with more than 200 countries. So this treaty lists some 900 species that cannot be commercially traded as live specimens or wildlife products because they are in danger of extinction. So actually, this treaty also restricts some of the international trade of 2,100 other species because they are endangered. But it also has some drawback. So they have the effect of this treaty is limited because the enforcement is difficult and it is convicted violators often pay only small fines, very small fines. Also, members of the member countries of this treaty, they can exempt themselves from protecting any listed species. You know what happened in China long back when the China met a very big drought, okay, and then the peoples they haunted the wildlife, and then they allowed it. Okay, so that's what okay, the reason being the China is the source of originating many type of zoonotic diseases because they're saying that okay, they're not purposely have done it, but the Wuhan market, okay, not only Wuhan market, many markets are there, which are because they have the tendency of trading the wildlife. They have liberated the rules. Okay, though it is a natural way of transmission of the diseases, but who is responsible for them? So that's the point here. So if you look at the conservation, okay, so you can, the classification of conservation, okay, because the conservation of the biodiversity it is very much important because biodiversity is one of the important tools for the sustainable development. So the enormous value of biodiversity, commercial, medical, genetic, aesthetic, and ecological importance, which will emphasize the need to conserve the biodiversity. So the conservation is defined as, you can define them, the management of biosphere, which will yield the greatest sustain a present generation while maintaining its potential to meet the needs of the future generation, not only the present generation. It has to maintain its potential to meet the needs of the future generation. That's the point here. Okay. So, what is the advantage or what is the need of conserving the biodiversity? Because it provides immediate benefit to the society, such as recreation and tourism, drugs, herbs, food, and other important raw materials which can be derived from the plants and animal sources. It also preserves the genetic diversity of the plants and animals. And then to ensure the sustainable utilization life supporting systems on the earth. And this may lead to conserve some of the essential ecological diversity and then the life supporting systems. Since the biodiversity loss, which will result in ecological and environmental deterioration, okay, what are the types or strategy to conserve the biodiversity? So we have classified into two major categories. That's what I have given here: in situ and ex situ conservation. Okay, so in situ means the protected area network. Okay, so we can classify them into different subcategories: the wildlife sanctuaries, biosphere reserves. Okay, even the biosphere reserves classified into terrestrial and marine. If you look at the ex situ, okay, home gardens, seed banks, gene banks, cryopreservation, botanical garden, arboreta, zoological garden, aquaria, and so on. So look at this, the in situ conservation. So the hotspot, biosphere reserve, national parks, wildlife sanctuary. Okay, so these are some approximate value. Okay, so which are considered as the in situ conservation. And then the ex situ conservation, botanical gardens, zoological park, wildlife safari parks, and then cryopreservation, tissue culture method, in vitro fertilization, seed bank, pollen bank, gene bank, field gene bank, aquaria, home garden. Okay, if you look at that, so the many of them you may know that. Okay, so I don't know how many of you know about the cryopreservation. If anyone know, now you can please switch on your mic and can you please share that. Do you have any idea of cryopreservation?
In absence of air, it will be present.
Sorry, it's not yeah.
It's a storing of genetic material under -70 degrees centigrade.
Yeah. Anyway, not exact, but anyway, close to my answer. Actually, it's not only storing of the genetic material. You can store the cells, the germ cells like sperms, eggs, and then even the ovarian tissue and all. Okay, and then -70. Since can you please precisely say the temperature of the cryopreservation, or on which is used to make this cryopreservation possible? Name of that?
So
Sir,
Yeah, please.
-196 nitrogen.
Yeah, exactly. Very good. May I know your name?
Yeah. Sir, Dr. Lakshman, I'm a veterinarian, sir. That's why I know this.
Excellent, sir. -196.
okay.
Yes,
Yeah,
sir.
which chemical we are using for that, sir? Anything?
Liquid nitrogen, is that?
Yes, actually. Yeah,
Yes,
good.
sir. Liquid nitrogen.
Yes, sir. Liquid nitrogen. Yeah. I think doctor may know very well because it's a routine procedure in animal husbandry and then the veterinary thing. Actually, most of the veterinary hospitals, okay, they have this facility. Okay. I think Dr. Raghu, please, can you please switch off your mic? Yeah, thank you so much. So the cryopreservation procedure, I will show you the video, okay, because some of the students and other personnel, those who are not aware about the cryopreservation, they may not about this. So it's just storing, okay, the long-term preservation of the necessary gametes, the germ cells, male or female germ cells, ovarian tissues, okay, and other samples. Okay, they have a cryopreservation tank. So the cryopreservation facility is very much needed. even to store your stem cell. Okay, so the cryopreservation is used to store the stem cell, because you may know that. Okay, so it's a procedure which is followed in the hospitals in the recent days, because a lot of stem cell bank have they have initiated. And then in vitro fertilization, okay, you know that. Okay, so all these things are coming under the ex-situ conservation. So I will show you in the forthcoming slide, and then I have a video for you. Okay, so in situ conservation. So this method involves the protection of the endangered species in their natural habitat. It helps in recovering the population in the surrounding where they have developed their distinct features. So the examples of all the nature reserves, wildlife sanctuaries, all these are coming into the in-situ conservation. Whereas the ex-situ conservation, it involves the placing of the threatened animals and plants in special care unit for their protection. It helps in recovering the population or preventing their extinction under the stimulated condition that is closely resemble their natural habitat. Okay, so all the botanical garden, zoological park, so all the things which I have listed previously is coming under ex-situ conservation. Okay, so it's just a detailed repetition. That means, so how it's conserved. So if you put it in in-situ conservation, so it will maintains the animal's normal behavior. Okay, because it won't affect the animal's behavior. Okay, and then the natural habitat also will be conserved by using this method. And then the food chain structure of the ecosystem will be maintained. Okay, because it's a components of the ecosystem. It has to be maintained in a proper way. It should not affect it. And then the captive breeding does not work for all the species. The habitat can be used for the reintroduction of animals. And then this habitat remains available to other species. So, one example, the Leighton Moss Reserve in the UK, they maintain some coastal marsh, which is ideal breeding ground for the avocet. And then the Project Tiger Reserves in India. So if you take the ex-situ conservation, that means keeping the animals away from its natural habitat. So this method will work if the habitat is lost, the original habitat is lost. And then by this method, it is easy to isolate the animals from the threats. The species may be too rare to breed to maintain the populations in the wild. And then the captive breeding can quickly increase the numbers for some species. So examples, the captive breeding of some pandas and some peregrine falcons. So examples, okay, for the ex-situ conservation we discussed in the previous slide. So if you look at the plant conservation, okay, so here again, if you classify them into two major categories, in situ and ex situ. So the tree diversity will be managed and monitored in natural habitats, whereas in the ex situ method, the tree diversity curated outside of its natural habitat. And then in situ, so there were research, and then horticulture, education, so maybe the purpose. And then in ex situ, germplasm, creation of germplasm, the seed bank, by using cryopreservation and the micropropagation techniques, and make some living plants, the conservation collections, reference collections, display specimens, and so on. So the advantage, so the species will have all the resources for which they have already adopted. So the species will continue to evolve in their own environment without any hustle. And then the species have more space. So the bigger breeding population can be kept here by using this method, in situ conservation method. It is cheaper to keep an organism in its natural habitat. So all these are some of the advantages of the in situ conservation. So then the ex situ, the animals maintained in zoos or wildlife parks, especially when their original geographic zone is not available, the captive breeding of animals. So this will work well for the species which are bred easily in captivity, like geese and deer. So the more specialized species are difficult to maintain here, but the animals isolated in captivity do not evolve with their environment because it's not its it may be resemble its original habitat, but not exact the original habitat. Small gene pool inbreeding, and then in vitro fertilization may be utilized. So it is possible to restore a population in a captivity. So as discussed, okay, so these are some methods which is adopted in exotic conservation. So now I'll show you a video. You just go through the video. Just a minute, I'll share my screen completely. So I hope now the video is visible. Yeah, actually, this is a cryobiology lab, which you call it as frozen zoo. Okay, so as I told you, so they are using to preserve DNA, sperms, embryos of the endangered species. Okay, to ensure their future. Okay, okay, even some ovarian tissues also can be stored here. So what they need a well-trained embryologist and then a set of doctors, maybe a gynecologist, and then veterinarians. I'm talking about the frozen soup. Okay. So here they have a setup like a cryopreservation unit. Okay. So here their job is to collect the samples, and then if needed they may go for the some artificial fertilization methods like in vitro fertilization, IVF. So many artificial fertilization methods are available. Okay, you just watch this video. Actually, this is the cryopreservation can. Actually, cryocan. It's called as cryocan. So, which will have a provision for handles, a lot of handles. So, at the end of the handles, they may have a ruler like collecting tubes. Okay. So, actually, this can is filled with liquid nitrogen. So, the liquid nitrogen have the temperature of minus 196, whereas the remaining empty portion within the can, that means above the liquid nitrogen, the empty can. So that's called vapor phase of the liquid nitrogen, which may maintain around 160, 150 to 160-degree temperature. So here the samples are going to be kept. So actually, this is the storage canister. Actually, here they are using. So depending upon the sample, they may use the different types of storage vessels. Like if it is eggs, okay, they may use some straws, okay, which are sterilized, and then the straws will be packed in such a way not to damage the egg which is present inside, and then they will label it, and then will seal it and label it with the straws. And then if it is sperms, and then they may use some cryo vials. Okay, the cryo vials, they put the sperms, and then they will add some additives like cryoprotective agents. Okay, you know, many cryoprotective agents are there. Examples: glycerol, DMO, so on and so on. And then they will seal it and then put it in this. Okay, so so what they have done, okay, I hope you may understand what they have done. So they have done nothing. Actually, they have by a team of doctors with the help of the embryologist, they have collected the eggs from the ovary. Okay, what they will do, they will go for the induced ovulation methods by giving some hormone injections. They will induce the ovulation, and then they will collect as many as possible eggs, and then the embryologist will identify the good quality of eggs. Okay, and then the embryologist, they will identify the good quality eggs and segregate them, and then they will remove the some unwanted debris around the eggs. Then they have already collected the sperms from the male animal, and that sperm will be allowed to inoculate. They will inoculate around the eggs, and then once the sperm fertilize the particular egg, and then they will observe under a microscope, and then they will find out the pronucleus formation. And once they find that the pronucleus formation, that's indication of the successful fertilization, and then the egg will start to divide, and then they will allow the egg to develop, either by putting it in the womb of the other animal or the same animal. Okay, so this is a method which they are using. Okay, so I just show you one video like this. Many videos are there. Okay, to make you to better understand about the procedure. Okay, so you just think that, okay, the cryopreservation method is not only for the animal, and then even for the plants they are using the cryopreservation methods. Okay, to store some long-term storage of seeds and embryonic axis and so on. Okay, so what are the advantages and disadvantages of the X-ray conservation methods? Okay, if you take a seed bank as an example, so it have an advantage of, okay, to propagules readily available for the users. The minimum work, little space will be required. Okay, and then the variations can be conserved. Okay, which provide the species range adequately sampled. Whereas the disadvantage of this method is, okay, so the recalcitrant seed cannot be conserved, and large seed preservation is problematic. Okay, so they does not conserve the associated species. To take the pollen bank, for that also the minimum space will be required. This will be applicable for plants with the recalcitrant or for the orthodox seeds. But the disadvantages, only half of the genome will be get conserved. And then the tricellular pollen storage is problematic here. And then if you look at that, DNA banks or gene bank, okay, so the minimum space is required, but species with high extinction potential can be conserved by using this. This can be applicable to all kinds of plants and animals. But what is the disadvantages of this gene bank method is it's very costly to maintain the system. That means not an easy method, because this needs some required skilled persons. Likewise, if you take the tissue culture bank, so here the minimum space will be required, and then the genetic erosion is reduced. So short time is required to produce the propagules. Disadvantages, sometimes the sampling is problematic. Okay, and then if you look at the cryopreservation of the sperm, ovum, or the embryo, so all kinds of plants and animals components can be preserved. But what is the disadvantages of this method is it involves freezing with liquid nitrogen. Okay, it's a very costly method. Okay, the viability of the healthy clone is sometimes questionable if they have not have a proper experienced skilled person, and then if they are not maintaining the system proper manner. Because you know very well. So cryopreservation means freezing of the sample at sub-zero temperature. Many methods are there in cryopreservation. Okay, so many methods are there. See here. One is slow schooling. Another one is stepwise cooling. Next one is vitrification. You know what will happen? What will happen if the freezing is done? Sometimes, because the cell membrane may get damaged or the cell organelles may get damaged, because during the freezing, the water molecules which is present in the cells may get frozen, and then ice crystal formation will happen. Ice crystal. This ice crystal, they may damage the membrane of the cell or the cell organelle. So that's why they have standardized, depending upon the sample type, they have standardized different methods of cooling. That means preservation, cryopreservation. If you take the slow cooling method, the advantages of this method is it has the stability from cracking, relatively non-toxic, because the cryoprotectants, that means cryoprotectants means the additives which you are adding, like glycerol, DMSO, and so on. These are called as cryoprotectants. They may take little time, okay, and then the disadvantages, it requires the equipment, because you need the proper equipment to go for it, because there will be a controllable freezer, okay. So like how we are setting a program in a PCR, likewise that there will be a programmer, okay, at what rate of temperature it has to be reduced, the temperature has to be reduced, okay, how much, 2 degree per minute or 5 degree per minute, like that, and we have to. So some equipment we needed, and then slow recovery. Okay. So, and then it can be low applicability with the tropical species. And next method is petrification. It's called fast freezing. The other name of petrification is called fast freezing or sudden freezing. Okay. So here, there are no special equipment is needed. It's a very fast procedure and fast recovery too. But petrification solutions are toxic to many plant samples. And then there is cracking, means that cell membrane damages. Damages may occur. But it requires some careful timing. Yeah. So likewise, another methods are there, encapsulation or dehydration. So here also no special equipment is needed, and they're using some non-toxic cryoprotectants. Simple thawing procedure they have applied. It requires some handling, okay, each bead several times, because some plants, they do not tolerate the high sucrose concentration. So that's another aspect. And then dormant bud preservation. So here it's easy and then useful for many temperate tree species. But the disadvantages is it requires freezing equipment, larger storage space, and then the recovery requires grafting or budding, and so on. So as I told you, that the temperature, how we have to apply, and then how they have to choose the method of freezing, whether they have to adopt the slow freezing or stepwise freezing or vitrification. In the stepwise freezing, what they will do, they will bring the samples from the normal room temperature up to 0 degree. Then after 0 degree, and then they will go for a slow cooling, stepwise cooling. Okay, at a control rate of freezing. Okay, so everything lies on not to allow the sample to go for either dehydration or ice crystal formation or making any damages. Okay, so there are so many national parks in India, Okay, so I just given in the map. Okay, so the Indian wildlife sanctuaries, and then in Bangalore Zoo recently the former Chief Minister Jayalalitha, okay, so she visited some of the cubs, which is and then she named also. Okay, so I think let us stop with this, because it's time up, and then we will continue in the next session, because we have discussed about the preservation method, conservation methods, and we need to discuss about the hotspots, and then what are the megadiversity okay, in the next session. Okay, now if you like to ask any questions, you please ask me now.
Excuse me, sir.
Yes.
Sir, this is Shravan.
Yes, sir. Tell me.
Sir, anything about lyophilization?
Yeah, lyophilization is nothing but it's called freeze drying. So, say for example, some samples, what we will do, it's called like to make them into a powdery form. Okay, because that will be easy to transport, without any contamination. Because if you want to have any samples, like primers which is used in recombinant DNA technology. Okay,
Yes, sir.
I'm just giving one example. Okay, actually, what they will do if you order the primer, okay, they will give it in a powdery form, in a lyophilized form. Okay, even some of the chemicals which is used in the molecular biology, they will give it in a lyophilized form. So the advantages of having lyophilized form is because we are getting its method is called freeze drying by using a lyophilizer. So just to avoid the contamination and then easy to transport. So once you received, okay, you can reconstitute the lyophilized form by using some sterile water, and then you can reconstitute, and then it will become a liquid form. Then you can aliquate, and then you can use it.
So as well, the microbial cultures also been transported in the same ways.
Microbial culture, I think no need of go for lyophilization, sir, because even a glycerol stock is well enough for microbes if it is a bacteria. Okay, if you want to store a bacteria, just assume that it's a E. coli. Okay, if you have a mother culture, then okay. Then usually the mother culture we will take very rarely, because we will put it in a stab culture, mother culture, and then whenever needed, you will take it, and then you will go for a broth culture. And then from the broth culture, if you have a good quality of broth, and then without any contamination, you can add equal volume of the glycerol and then mix it very well, and then seal it, and then keep it in a -20 freezer or -70 freezer. That's enough. Okay, that will be good for transportation.
Some of the culture banks, microbial banks, they ask for lyophilized cultures.
Okay.
Like when we'll be registering the new microbe or anything, like they ask for these lyophilized tubes.
Okay. Okay. So microbes
So
in the sense which sample? Because we used to call bacteria as a microbe, virus as a microbe, fungi also, which is
yeah,
which
so
for which sample they are asking for that.
yeah, so for microbial culture itself, sir, like bacteria.
Oh, I see.
Bacterial cultures. Generally, when we wish to publish or any new species, and then those gene banks, they ask for these microbial culture centers. They ask
Oh,
for lyophilized tubes.
okay.
Uh,
What they will do? They will get it from you, the sample, lyophilized form of the
yes, sir.
bacteria.
Yes, sir. We have when we have to get the registered number.
Okay.
Before we go for publication, we have to get that number of the culture.
Okay.
So we have to deposit it, those bacterial culture in two of different microbial culture centers
Okay.
in any part of the world.
Oh, I see. Mhm.
So for that, and even the transportation of that.
Yeah, that's the purpose, because you are going to
Yeah.
transport it. No, it's a
Yes, yeah.
so without any go to the distance location. That's why.
So, and the lyophilized tubes also will be very thin, and you can send it in a bulk, uh, packed in a compact packed, and can be sent in.
Yes, you're right. Actually, it will minimize the damages also, because
Yes, sir.
even in glycerol stock, okay, you can go for a glycerol stock, okay, up to for a few years, and then after that it will get contaminated if you're not
Correct.
properly maintain your freezer. Anyway, the
Yeah.
lifeless form is always safe.
Yes, sir. Yes, sir.
Thanks for the nice information, sir.
Thanks, sir. My pleasure.
Okay, so now I would like to post the attendance link in the chat box. Okay, so I request you all, you please fill the attendance link, and then hopefully we will meet again in the next session after this holidays. So at that time we will discuss about the further topics of your syllabus. Okay, so I wish you all to take care, and then